If you've ever pictured, ancient DNA, maybe resurrected dinosaurs, or just a world perfectly frozen in time, chances are amber comes to mind. That iconic mosquito from Jurassic Park. It totally captured our collective imagination, didn't it? Made fossilized tree resin feel like, well, pure magic. But way before Hollywood, amber fascinated people for millennia. You see it in stunning jewelry, amazing art. Some cultures even gave it mystical powers. Today, we're doing a deep dive into this incredible world of amber, not just because it's beautiful, but because it's this unparalleled natural time capsule. We're going to explore the amazing insights it gives us into prehistoric life. From the surprising places it's found all over the globe to the really cutting-edge science revealing its deepest secrets. Our mission really is to understand the huge biodiversity amber preserves and the, frankly, ingenious ways scientists are unlocking its ancient mysteries.
It's true. Jurassic Park definitely sparked a huge global fascination. But the actual science, I'd say, is even more astonishing. What makes amber so unique is the fidelity, the sheer detail with which it captures life. Unlike other fossils, imprints or mineral replacements, amber can preserve organisms with lifelike, three-dimensional detail. It's a remarkably distinct window.
Right, a clearer picture in many ways.
Exactly. Often much more revealing about ancient ecosystems.
So, building on that, we often just think amber like it's one thing, but it's actually found in nearly 200 known deposits around the world. Some are incredibly old, stretching back to the mid-Carboniferous. But what's fascinating and maybe a bit surprising is that only a relatively small number of these deposits actually have abundant biological inclusions. And those are mostly from the Tertiary or Cretaceous periods.
Yeah, and what's striking is how these different deposits, even though they were formed by, different types of trees, maybe under slightly different conditions, they still offer broadly comparable ecological insights. Well, it's mostly down to the trapping mechanism. Tree resin is sticky stuff, right? It works pretty much the same way regardless of the tree. But that said, each deposit definitely has its own unique personality, you could say, its own special finds.
Okay, let's unpack that. Maybe walk us through a few key examples. Give us a sense of this global picture.
Sure. Let's start with Dominican Republic amber. That's from the Miocene epoch. People only really started studying it seriously in the 1960s, but it already boasts over 1,000 described fossil species, and the preservation is often just exquisite.
Wow, 1,000 species just from there.
Yeah, and one really intriguing find is a fossil termite, Mastotermes electrodominicus.
Okay, what's special about it?
Its closest living relatives today. They're only found in Australia.
Australia. But this is Dominican amber.
Exactly. It hints at these deep, ancient biogeographical connections. Continents moving, species getting isolated, long before things settled down like they are now.
That's fascinating. Okay, what about another famous one?
Well, there's Baltic amber. Eocene age from northern Europe. This is probably the most famous, the longest studied deposit. Over 3,000 described species, mostly arthropods, insects, spiders, that kind of thing.
3,000. But I sense a but coming.
There is, yeah. A significant challenge with Baltic amber is that many of the early descriptions from back in the 1800s, well, they're just not adequate by modern scientific standards.
Right, science moves on.
It does. And worse, many of the original specimens, those descriptions, were based on the type specimens, which are absolutely crucial reference points. They've been lost over time. Wars, poor curation, you name it.
Ah, that's a real shame for research.
It really is. It makes reevaluating those early findings very difficult.
Okay, let's maybe shift continents. You mentioned Indian amber.
Yes, from Vestan in Gujarat. This is a relatively recent discovery, only found this century. And its significance is huge because it formed before the Indian and Asian tectonic plates collided.
Oh, right, that massive geological event around 50 million years ago.
Precisely. So this amber gives us an unparalleled snapshot of life and how continents were arranged before that huge rearrangement happened.
Yeah.
Truly unique paleobiogeographic information.
Incredible timing. And wasn't there another really old one?
Lebanese amber. That, along with some finds from Jordan, represents the oldest Cretaceous amber that has really diverse biological inclusions.
Cretaceous.
Literally.
So dinosaur times.
We're getting into that era, yes. And what's remarkable here is how it documents the diversification of modern types of arthropods and also the disappearance of some older, more archaic groups. And this aligns really nicely with the rise of flowering plants, the angiosperms, and whole new ecosystems emerging.
So it's like watching evolution happen in resin.
In a way, yes. And even more captivating, Lebanese amber preserves some of the oldest specific behaviors caught in the fossil record.
Behaviors? Like what?
We're talking direct evidence. Mating in certain types of flies, mite parasitism on midges,
Yeah.
Caught right in the act millions of years ago.
Wow. Just frozen moments.
Exactly. Frozen behaviors, as one source calls it.
So, thinking about all these inclusions, what really stand out? It's not just the variety, is it? It's these interactions.
Absolutely. It's not just who lived, but how they lived.
Things like mating, as you said, maybe mate guarding, predation. I read about a larva disguised with bits of other insects.
Yes. Like a little predator wearing camouflage made of its victims, ant bits, spider bits, pseudoscorpion carcasses. It's amazing detail.
So is it fair to say amber offers this crucial window into the actual, dramas playing out in ancient ecosystems?
That's precisely it. These fossils are vital for both quantitative and qualitative ecological studies. They help us see the impact of big events like mass extinctions, what was life like just before or just after.
Mm.
They're also key for studying predator-prey coevolution over huge timescales and understanding the rise of flowering plants and their pollinators, a massive shift in Earth's history.
And you mentioned phylogeny, mapping evolutionary relationships.
Yes, invaluable data for that. And for understanding past biogeography, like those disjunct distributions we talked about, the Dominican termites and their Aussie relatives, it paints a picture of a changing planet.
And it's not just insects, right?
Oh, not at all. The diversity is staggering. Microscopic stuff like bacteria, fungi, plant bits like leaves, pollen, even small vertebrates, frogs, lizards, bird feathers, eggshells.
Really? Actual vertebrates.
Yep. And on rare occasions, even tiny mammals. We've even found disease vectors preserved, like microscopic trypanosome parasites, right inside their sandfly hosts.
That's mind-blowing detail.
It truly is.
So with all this incredible scientific value, these things must be valuable in other ways, too.
Oh, absolutely. Especially in the 90s, after Jurassic Park, the market for amber with inclusions just boomed.
I bet. But booms often come with downsides.
They do. That boom saw a decline partly because of widespread fakery, and also copal, which is younger, subfossilized resin, not true amber, being passed off as the real deal.
Ah, the forgery problem. That brings up a critical point. How do we know it's real? Especially if fakes can be sophisticated.
That's a really important question. Authenticity.
Mm.
Forgeries are incredibly common. They can have serious financial impacts, obviously, but also major scientific ones if fake data gets into the literature.
And just buying from the source isn't enough.
No guarantee at all. Even buying directly from miners doesn't ensure it's genuine amber or that the inclusion wasn't, added later.
Okay, but thankfully there are ways to tell. Methods to distinguish real from fake.
Yes, definitely. There are several practical methods, starting with some simple physical tests anyone can try.
Like what?
Well, UV light is a good start. Genuine amber usually fluoresces, often blue or yellow, while most plastics used in fakes don't. Then there's the flotation test. Amber and copal are light. They'll float in a concentrated salt solution. Most plastics are denser and will sink.
Okay, simple enough. What else?
A hot needle test. Touch a red-hot needle, point to an inconspicuous spot. Real resin gives off a characteristic piney, resinous smell. Plastics. You get that acrid, burning plastic smell. Pretty obvious.
Right, and scratching it.
Yep. The surface scratch test. If you carefully scratch genuine amber, it produces a fine powder. Plastic tends to peel off in a sort of coiled thread.
Useful tips. Are there more definitive tests?
For sure, there are chemical tests. A drop of ethanol or ether on copal will make the surface tacky. Some plastics react to genuine amber. Generally, its surface stays unchanged. And for absolute scientific certainty, labs use techniques like infrared spectroscopy. That gives a unique chemical fingerprint of the resin. Or pyrolysis gas chromatography, which basically breaks down the material at a molecular level to identify exactly what it's made of.
Very high-tech. But you also mentioned visual clues being important.
Hugely important, often the most telling. Remember those frozen behaviors? We'll think death throes. Genuine inclusions often show signs of struggle. Jumbled wings, tangled legs, maybe even tiny fossilized blood droplets from the struggling insect.
So a perfectly preserved, symmetrical insect might be suspicious.
Immediately suspicious, yes. Nature isn't usually that neat, especially when you're drowning in sticky resin. Another big red flag is often a thin air layer you can see between the organism and the surrounding material in fakes. Synthetic resins don't wet the surface the same way natural resin did.
And people have actually faked famous specimens.
Oh yes, there have been some notorious examples. The behelmet frog in the American Museum of Natural History, the Piltdown fly in London's Natural History Museum. These involved expertly inserting modern organisms into genuine pieces of amber. Very deceptive.
Wow. Okay, so diligence is key. Once we are sure a piece is genuine, how do scientists actually start studying these tiny things? Seems like a massive challenge looking through potentially cloudy, irregular amber.
It is a challenge, and preparation is absolutely critical. First, you need to understand a bit about the optics. Amber is usually transparent or translucent. The clearer it is, the less light gets absorbed. Inclusions are visible because they have different opacities, different refractive indices.
Refractive index, like how light bends when it goes through water.
Exactly. So when light hits a curved or scratched amber surface, it bends unevenly, distorting the view of what's inside. The whole goal of preparation is to create a perfectly flat, optically polished surface with as little amber as possible between your eye or the microscope and the inclusion.
Makes sense. So, what are the first steps when a promising piece comes into the lab?
Usually it starts with cleaning the raw piece. Sometimes just smearing a bit of oil on the surface fills tiny surface scratches and can reveal hidden inclusions you couldn't see before.
A simple trick.
Yeah. If something interesting pops up, then more involved prep begins. Typically, cutting and grinding. You'd use a circular trim saw with a very thin diamond blade. Water is used as a coolant, vital to stop the amber overheating and cracking. After trimming away excess amber, the surface around the inclusion is ground down. This can be done by hand or on a machine called a flat lap, using progressively finer grades of abrasive paper, like starting coarse and going down to superfine.
Constantly checking, I assume.
Yeah. Constantly checking under a microscope. You really don't want to grind away part of the fossil. Then finally, polishing, usually with a fine-apt pad and diamond compound or alumina powder to get that mirror-smooth optical surface.
Sounds painstaking.
It is. And you have to be incredibly careful. Amber can have internal fractures, and the stress of cutting or grinding can easily cause a precious specimen to break.
Okay, that's the standard process. But what about those tricky situations? Really brittle amber or incredibly tiny, delicate inclusions where cutting is just too risky. Are there special techniques?
Oh, absolutely. For enhancing visibility, especially if the surface isn't perfect, immersion fluids are great. You trim the piece roughly, then immerse it in a fluid with a refractive index very close to amber's. Cedarwood oil is a classic example.
And that helps how?
It dramatically reduces light scattering off the surface, eliminates distortions from minor scratches or irregularities you can see much more clearly, and by rotating the specimen in the oil, you can view the inclusion from multiple angles.
Clever. What about really fragile stuff, like that Lebanese amber you mentioned?
Right, for particularly brittle ambers like that, where large pieces often shatter, the Canada balsam technique is invaluable. Instead of cutting, you carefully shave off very thin slivers of the amber with a sharp razor blade, getting as close to the inclusion as possible without touching it. Then, you mount this thin sliver on a microscope slide in a fluid mount, slowly adding Canada balsam.
Which also has a similar refractive index.
Exactly. The balsam seeps into any tiny surface cracks in the amber sliver, making them optically disappear, improving clarity massively.
Neat. Any other tricks for brittle pieces?
Another approach is synthetic plastic mounting. You can embed the entire fragile amber piece within a block of clear plastic. This makes it much easier and safer to handle, prevents it shattering during grinding and polishing, and you can still polish different faces of the block to view the inclusion from various angles. Plus, it protects from oxidation over time.
That seems really practical. You also mentioned something controversial, dissolving the amber.
Ah, yes. That's been attempted, particularly with Lebanese amber, using solvents like chloroform to actually dissolve the amber matrix away from the inclusion.
Wait, destroy the amber to save the fossil?
Seems counterintuitive, right? But for some paleontologists, especially those focused purely on the anatomy of the insect itself, the goal is to extract it. If successful, you can recover articulated fragments, sometimes still soft, almost like freshly collected specimens. It allows for direct anatomical study you couldn't do otherwise. But yes, it's destructive to the amber itself.
No, definitely a trade-off. So, okay, the piece is prepped, maybe using one of these special techniques. What tools do scientists then use to really unlock the secrets inside, things invisible to the naked eye?
The absolute workhorse is the stereo microscope. It's essential for this kind of work
Why stereo specifically?
because it provides a larger depth of field compared to a standard compound microscope, and crucially, it produces a true three-dimensional image. You need that 3D view to understand the structure of these preserved organisms. Compound microscopes are designed more for flat, thin slices.
And getting the light right must be key.
Absolutely crucial. Flexible lighting systems are essential. You need light from below, transmitted, light from above, incident, sometimes hitting it from different angles. and a technique called dark ground illumination is often used. It lights the specimen against a dark background, making very fine details like tiny hairs on an insect's leg really stand out.
Makes sense. What about taking pictures? I imagine getting everything in focus is tough if the fossil has depth.
That's a major challenge. Often the inclusion is much thicker than the microscope's depth of field at high magnification, so only a thin slice is in focus at any one time.
So how do they get those amazing, fully sharp photos we see?
The answer is digital image stacking. You take multiple digital photos, maybe dozens, maybe hundreds, at slightly different focal planes, moving through the entire depth of the specimen. Then specialized software takes this stack of images and digitally combines the sharpest parts of each one into a single composite image where everything is perfectly in focus.
That's brilliant technology solving the problem.
Exactly. It revolutionized amber fossil photography.
Okay, that's amazing for visual stuff. But I've heard whispers of even more advanced techniques, almost sci-fi ways to see inside the fossils without even touching them. What's on the cutting edge?
You're right. Things have advanced incredibly. High-resolution X-ray computed tomography, or HRCT, is being used with fantastic results.
Like a medical CT scan, but for fossils.
Precisely, but much higher resolution, down to the micrometer scale. It's non-destructive, which is a huge plus. It generates incredibly detailed 3D reconstructions of the inclusion. You can digitally slice through it, view internal organs, muscles, things you could never see otherwise.
Wow! So you can basically do a digital dissection.
Exactly. It's especially useful for things like Baltic amber, where inclusions are often coated in that milky white emulsion, obscuring surface details. CT can see right through that.
Incredible. Is that the ultimate tool now?
It's amazing, but there's even propagation phase contrast X-ray synchrotron imaging.
That sounds intense.
It is. It uses similar principles to CT, but employs the extremely bright focused X-ray beams generated by a synchrotron particle accelerator.
Okay, why do you need that kind of power?
Think of the difference between a regular flashlight beam spreading out and a laser pointer, a single intense beam. The synchrotron provides that laser pointer equivalent for X-rays. This intense, single-wavelength beam is incredibly good at penetrating even completely opaque amber, allowing scientists to screen large, murky samples for hidden inclusions that would otherwise be missed.
So you can find things you didn't even know were there.
Exactly, and the data quality is phenomenal. You can get such detailed 3D models from this data that you can actually feed it into a 3D printer.
You're kidding. Print a model of the fossil.
Not kidding. Scientists can create a physical and large plastic model of the inclusion based on the synchrotron scan data. They can hold in their hand a perfect replica of an ancient creature they've never actually seen directly.
That is absolutely mind-bending. So what does all this tech mean for the future of understanding ancient life through amber?
Well, these modern techniques, they're expensive, they need specialized facilities, sure, but they're proving to be incredibly powerful complements to the traditional methods, not replacements. They're dramatically expanding what we can learn, showing that even tiny, previously overlooked amber fossils can provide a huge amount of evolutionary information, what we call phylogenetic inference. It's directly challenging views held by some biologists working only on living things who might have dismissed fossils as too incomplete. Amber fossils, studied this way, are proving indispensable. They're unlocking ancient ecosystems in ways we never thought possible.
So we've really journeyed deep into this world of amber fossils today. From the different deposits scattered across the globe, each with its own story, to the meticulous preparation and now these incredible high-tech tools. It's truly a remarkable natural archive, isn't it? Capturing moments in time with such, well, lifelike fidelity.
It really is. And thinking about the bigger picture, it really underscores the ongoing importance of looking after these time capsules. Because despite being ancient, amber isn't indestructible. It does deteriorate over time, mainly due to oxidation. It can darken, get crusty, develop cracks.
Oh, so conservation is an issue.
Absolutely. Proper conservation and curation in museums, and even for private collectors, is vital. And having a good recording system linking the physical specimen to high-quality digital photos, scan data, all the information is essential to preserve not just the amber, but the invaluable scientific knowledge it holds for future generations.
That's a really important point. So for you, the listener, what's the takeaway? Amber isn't just a pretty stone or, a plot device from a movie. It's this amazing testament to the power of nature to preserve and the ingenuity of science to uncover. It's a constant reminder that the past is always revealing new secrets, just waiting for us to find new ways to look. And maybe here's a final provocative thought for you to mull over. Imagine if, someday, We could examine every single microscopic fossil, every captured behavior, and every single piece of amber from every deposit worldwide using technologies we can't even conceive of yet. How might that fundamentally change our understanding of evolution, of past climates, maybe even the intricate interconnectedness of all life on Earth across millions of years? What secrets are still locked away?